Assalamu alaikum. My name is Bareera Khan and I'm a final year student of Dow Medical College. My topic today for this session is synergetic therapeutic events of Magnifica indica and Azaratheca indica and diabetes associated atherosclerosis. This is a rat model study which is a preclinical trial study done at our university. Our main authors are Dr. Anush Tahir, which is our primary supervisor. My group members are Nashra Mughal, Aisha Masood, Sumaya Farooq, Sahar Bareera, and our co-supervisors are Dr. Adnan from KU and Ms. Dr. Maryam Mola Baksh. We have our general acknowledgement for Dr. Khurram from Animal Farm and Mr. Irfan and Mr. Haji from Animal House for the assistance. We have associated with Dow Medical University of the Medical College and KU Department of Pharmacology. Appreciation for our principal, Dr. Sameer Qureshi, for giving us this research-driven environment and providing continuous encouragement to make this study possible. So let's move forward. Why do we need this study? The background of this study is that we have rising global burden of diabetes that will project into 783 million by 2045. This is also linked to a major cardiac disease called atherosclerosis driven by oxidative stress. So the limitations for these conventional treatments are basically that statins, ACE and anticoagulants, they are good alternatives, but the thing is they have major side effects that can cause muscle pain, liver toxicity and bleeding risk. Why do we move forward towards this study? Because the growing interest in herbal medicine. So this is why Azaratheca indica and Magnifica indica have flavonoids, phenolic acids and carotenoids that will move forward to a herbal extract treatment. The statement of the basic problem is that diabetes has disrupting insulin function, which triggers metabolical imbalances that promote to vascular inflammation that leads to atherosclerosis. So basically, alloxin-induced rats were used for this pathological changes to make the study possible for a human containing diabetes to mimic in rats basically. So the rationale of the study is that diabetes and atherosclerosis will affect 60% of Asia's population risk. Our current treatments have adverse side effects. So our research basically highlights that the combined extract treatment of Magnifica indica and Azaratheca indica is a safer and a natural alternative to produce those with atherosclerotic diabetes. So the main objectives were to combine the synergetic effects of Azaratheca indica and Magnifica indica. The second was to investigate the antioxidant and anti-inflammatory potential and to evaluate the association between alloxane, diabetes and the progression of atherosclerosis. So the materials and methods that were used for randomized controlled trials, which were from female albino rats. This was from Animal Farm, from our university. They were divided into 4 major groups: control, negative, diabetic atherosclerotic, treatment and combined treatment. So an extract was given. First they were ingested with the alloxane to produce diabetic condition, and then they were led to atherosclerotic, and then they were treatment with the treatment. So basically one of the groups, they had the treatment, other groups, they did not have the treatment to analyze the effects. So the diabetes were induced and overnight fasting, just like major human trials, and high-fat diet was induced for atherosclerosis. After the model induction, the treatment was given after 60 days and then tested for hormonal, biological and biopathological results. So the ethical consideration, the ethical consideration was approval IBC KU 4421-2024. They were used with immediate care and laboratory. Animals were strictly endorsed to accordance to international standards so that no animals were harmed during this entire process. So the results were formed through SPSS analysis and one-way ANOVA that produced that blood weight, body weight, blood glucose, HbA1c and cholesterol, they all were infected between treated and non-treated groups. So the histopathological evaluation were marked improvements in tissue agriculture. So we'll see the histopathological So first we have the histopathology of kidney. This was also done by the students itself. So first in the A1 we can see the glomeruli rat, which is the basic rat. In the B the tissues were destroyed. The C1 they are without treatment and D1 with treatment. Same with histopathology of result. The combined extract shows an improved hemorrhage of liver cells of diabetic atherosclerotic treatment with our treatment. This is the pancreas, that Langerhans of control rats. There was seen shrinkage in between the C1s and the B1s, but in our treatment, we can see that they were intact. In the histopathology of abdominal aorta of the heart, which is basically the main center where atherosclerosis is induced, so we can see that they are also treated and we have our aorta fully intact. So, and this is the artery. So in the A artery, it's without. In the B artery, you can see digression. And in the C, it's completely decreased lipid deposition. And in the D, where with treatment, they are fully intact. So the discussion and the analysis is that it reduces oxidative stress, and then it also reduces antioxidant and anti-inflammatory health. It has a fewer side effects. It's a herbal approach to newer and safe and cost-effective to the newer and more trendier medicines and expensive medicines in the market. So the conclusion is that the combined extracts of Azadirachta indica and Magnifera indica promise us antioxidants and anti-inflammatory effects of the diabetic model. These preliminary findings were an effective alternative to the conventional treatments in the market. So the recommendations that I would like to give to the presence of the entire team is that we should expand our clinical research that we did on rats to human trials to establish the clinical accessibility in herbal extracts, and in-depth molecular investigation should be done so these compounds can be figured out which of the combined extracts may reveal novel therapeutic targets for diabetes-related vascular diseases. Thank you so much.